to welcome everyone to the quarterly HCA Town Hall. And to make it easier for people across time zones, we actually hold two identical sessions this time. This is the second session. Some people have already heard the first one. I was not on it, it was Sarah. So I'm going to lead this session together with our Vice Chair of the Organizing Committee, Chloe Villani, and the HCA Global Coordination Lead, Shyam Prabhakar. And so if we can have the next slide. Let me tell you a little bit about what we're going to cover today. I'm going to tell you about recent and upcoming HCA activities. Then we're going to talk about the goals and requirements for completing the first version of HCA. Chloe is going to present the recently approved HCA Immune Atlas. And then we're going to cover our vision for HCA 2.0, including some of the landscaping analysis that we've done and the next steps that are coming up. And then, of course, the main goal of town halls is for us to have a discussion together. So we really encourage people's participation. And if you can submit your questions and your comments in the Q&A section at any time, and then we're going to try and address questions as we go along. And we reserve time at the end for a full Q&A. Okay, so if we can have the next slide first. Next. And so, I think most people think about time, there's, the academic year, there's the new year in January, there's many different ways. You can follow the solar calendar or the lunar calendar, but in HCA, the year is defined by the general meeting. So we're very grateful to everyone who joined the 2025 general meeting, which was in Singapore, and to Shyam, who's one of our hosts there this past June. And we're very much looking forward to welcoming you to the 2026 general meeting that will be in Boston next June. And it's going to be very special for us because it's going to be our 10th anniversary, our 10th year. And so let's take a little bit of a look at the year ahead in the next slide. So HCA currently has 18 biological networks, and at the time of the previous general meeting, we reached the midpoint of assembling the first version of the atlas, and we had draft atlases for 9 of the 18 biological networks. Our next goal, our next big milestone is going to be to have all 18 first version atlases to be completed by the time that we meet in our very special meeting in Boston next June. At the same time, we're really preparing for the next phase of HCA. We're approaching our 10th anniversary, and the ambitions have really grown and become both broader and more global. So back at the time that HCA began in 2016, when we had our first planning meetings, it was the time that it was just becoming possible to profile, say, tens of thousands of cells by single-cell RNA-seq. Droplet-based sequencing just came on the stage a few months earlier. But even then, we set our sights on having tens of millions or hundreds of millions, and kind of in the ultimate level, 1 billion cells or so in our first version of the atlas. Now, these kinds of numbers looked very far-fetched at the time, but they're completely the reality today, both because of advances in single-cell genomics, but especially because of advances in spatial technologies. And that means that we can now accelerate the expansion of the atlas with more data integrated that comes across more modalities, more tissues, organ systems, and to do it in 2 and 3 dimensions. Now, just as important is expanding the diversity of the atlas so that it reflects the breadth of human biology. And ultimately, our goal is to have a reference atlas that advances how we understand and treat disease, which is to fulfill our mission of the Human Cell Atlas. So later today, Chloe and Shyam are going to share results from the HCA Disease Focus Survey, which many of you contributed to. And that feedback has been invaluable, so stay tuned. We're going to come back to this. Now, what comes next? Next slide. Okay, so between now and June of next year, there's a lot of important work for us to do. And I want you to think of this as a collective call to action for all of you in terms of the ways that each of you can contribute and how important it is that each of you will contribute. So you might be contacted by a biological network coordinator in order to provide data or metadata. The data ecosystem team has really streamlined the ingestion process and they can guide you through it. So please respond to these requests. If you provide your data and your metadata, then your data can be part of the atlas. A second important thing is if you're like to and are willing to participate in community-driven cell annotation, there's going to be an opportunity for you to join the annotation jamborees on the cell annotation platform, or the CAP, which are organized by biological networks. And again, it's a way to contribute to your expertise and make sure that cells are actually called by the names you want them to be called, and it is the right cells that are called by those names. And then finally, if we look ahead, you can also get involved, of course, in the efforts for HCA 2.0, especially through the Spatial Omics Task Force. Now, please keep your HCA membership profile updated so it has your current email and your research interest, because this is what ensures that you're going to receive all the notices, all the emails, all the communications, and don't miss out on any opportunities. And later in the town hall, we're going to share more on how to stay connected. So a little bit, next slide, please. Thank you. So I already mentioned that we're going to have the HCA general meeting in Boston. This will be in June 16th to 18th, 2026, and it will be held in the Harvard Medical School Conference Center in Longwood. This meeting will be a hybrid meeting. The registration is already open. You see a nice QR code there. You can use it. There's also many other ways to register. We really hope to see many of you there, either in person or virtually. But it's not the only meeting I want to announce. So if you go to the next slide, we're also very excited to announce our future HCA Asia meeting, both for 2026 and for 2027. So in 2026, we're going to meet in Perth, Australia, and in 2027 in Jeju in South Korea. And there will be more information soon on exact dates and registration details, so just stay tuned. But you can already see these are two absolutely gorgeous places to go to. And then if we go to the next slide, these meetings really require a lot of resources to put out, and we're very, very grateful for both for our past and our future sponsors for these meetings. I especially want to call out 10x Genomics because it is a foundational sponsor for the 2026 general meeting, which will allow us to make this meeting very special. If you are actually interested in sponsorship opportunities, please contact us at meetings@humancellatlas.org. Next slide, please. Now, there's many different kinds of partnerships for HCA, and one of the ones that we shared in the March town hall and in the meeting in Singapore is our new partnership with UNESCO. And together with UNESCO, we identified strategic priority areas that really align with both HCA's mission and UNESCO's mission, and we're really proud to join forces. This will allow us to provide better for open science and equitable data sharing, to make sure that we have the right policy dialogues and ethical frameworks, that we are building the right capacity and inclusion for the different regions around the world, and that we are integrating and synergizing with other initiatives, other scientific initiatives. So this partnership with UNESCO will really help us do those things. And if we go to the next slide, in the months ahead, you're going to start seeing several joint initiatives that are planned, and they include an exploratory webinar series on single-cell genomics and the exposome, on our joint bioethics working group, on workshops to build extra capacity for single-cell genomics, and ways to support open science, science diplomacy, and public engagement. And we're going to keep you updated through our website events and our news pages and the newsletters and the social media so you can engage as well. Next slide, please. So now I'm going to move to the next topic on our agenda, switching gears to the science of HCA, and talk about the completion, or what is ahead of us in order to complete HCA 1.0. And for this, I want to take a closer look at our goals and the requirements for reaching this important milestone. So in the next slide, I want to start first of all with gratitude and congratulations to the team that completed their first version atlases and recently received approval from the HCA organizing committee. It's kind of cool. I mean, I think you all remember the lung. We have the lung, the brain, the eye, the organoids, developmental, the gut, the liver, and more. I won't enumerate all of them. 2 are up for organizing committee review in November, and the remaining 8 will be approved in 2026. So we're passing the halfway point for completion and plan to complete by the end of the year. Now, the most recently approved are the kidney, the gut, the development, the immune, and the liver cell atlas. Chloe is going to tell you all about the immune one, and it's very exciting. They will soon be available through the HCA data portal. The kidney atlas integrated and annotated 18 single nucleus and single cell data set. That's 1/8. And the resulting atlas contains 1.7 million single cell and nuclei, and they represent both healthy states and disease condition like acute and chronic kidney injury, which affect a lot of people around the world. And then the gut cell atlas has 1.5 million cells from 27 data sets and 288 donors. And that includes data from the Crohn's Gut Atlas groups supported by the Helmsley Charitable Trust. And this remarkable effort brought together 12 labs for annotation, which is a true testament to the strength of the HCA collaborative spirit. Then the Human Developmental Atlas includes more than 3.6 million cells from 188 donors. And this team integrated developmental data from 21 organs between 4 to 22 post-conception weeks. And that includes both published data, but also newly generated whole embryo data. And as I said, you're going to hear more about the Immune Atlas later today. And that includes a collaboration between the Immune Biological Network and the Genetic Diversity Network. This is going to be our most genetically diverse atlas. Over the next few months, oral and craniofacial and pancreas are coming up for approval in November organizing committee, and we'll continue to update you as more and more atlases are accepted and become available. And again, I want to thank all the teams and the leaders and the bionetwork coordinators for all the work across the world to make these efforts happen, from collecting the data to putting it together and annotating it and really making it available for everyone else in this coherent, integrated form. Each of these atlases is years and years of community-driven work and very shared commitment. So if we go to the next slide, I think it's useful to remember that each of these atlases follows particular guidelines, and that would make one of the things that makes them very different than just an aggregation of a lot of data sets that are out there. And so in order for an atlas to be accepted by the organizing committee, it has to show that it is robust to technical factors, that it incorporates landmark studies, that it is driven by the biological network, so it was done by the experts for the community, that it is annotated with expert input, and that the annotations and the supporting evidence are available in the cell annotation platform, the CAP, and that it has integrated data, metadata, relevant code, and all are publicly available. And so these approved atlases are made publicly available through the portal, and we keep an eye on our social media channels for all of those important updates. So next slide, please. So I think I started touching on what makes the Cell Atlas different and what sets it apart from other initiatives that are building tissue or organ-specific atlases. And part of it is the sheer scale, not just the scale of the data, but the scale of the scientists that participate, which means that we integrate a lot of expert views and the whole community. The commitment to diversity, so that the atlases are truly representative of human diversity, that the scope is the full human body and not just one particular tissue or part of a tissue or part of an organ, and that allows also cross-tissue analysis. That on the one hand, we are flexible, so we leave room for all the biological nuances, but we are aligned across the atlases, that we apply our methods very rigorously, and they were created and tested by the 18 bionetworks, and that we are participatory. Anyone can participate, anyone can be involved, and people from countries all over the world are part of HCA. Now, if you go to the next slide, sorry. To emphasize really the importance of the collaborative work that is needed to achieve HCA 1.0 completion, I think it's important to look at the 4 main requirements for completion, and that each of them depends on community input, and each of them is driven by the biological network. That includes that all the data and metadata are available on the HCA data repository and on CAP. This is important because it means that we will be able to enable cross-tissue integration and generate unified cell maps. The importance that we have, the requirement that we have all the annotation metadata on the HCA cell annotation platform is crucial because it means that we can achieve consensus in cell type annotations. We achieve transparency in the annotation so people understand why each cell type is annotated in the way that it is. And it allows the development of HCA gene panels for each tissue to be used by different types of methods. The fact that we require a full collection of tier 2 metadata for each of the biological network means that it should be possible to translate the atlases into new biological and medical insights that crucially depend on analyzing the data in light of these tier 2 metadata. And the fact that we require a more sustainable HCA data ecosystem means that we can support later on atlas expansion, drug discovery or discovery of new diagnostics, and are able to train or enable the community to train foundation models that really rely crucially on very, very large scale data sets. Next slide. Okay. I think you all remember the big publication package that we had actually about a year ago. We are very excited to announce the preparation of the next coordinated publication package, which will coincide with the 10-year anniversary of the HCA and the completion of version 1. This collection will highlight the work of the individual biological networks in creating the first version of the atlases, and will be an opportunity to publish the source dataset, to showcase the computational tools and the methods that we used in version 1, to highlight the impact of the first draft on the atlases, both in advancing biological discovery and in informing clinical and translational research. And your biological network coordinators will help guide and curate this 2026 publication package. In terms of the timeline, draft abstracts will be due from the biological networks in November 2025. We expect the submission deadline to be in March 2026, so that the final draft of the perspectives and other contents will need to be available in July 2026, and then the target publication date will be October 2026, which will be our 10th anniversary of kicking the planning process for HCA, which was October 2016. And so if we go to the next slide, this is my opportunity to ask you to join me in welcoming Chloe Villani, our vice chair of the organizing committee, and she's going to present the recently approved version 1 Immune Atlas. If you have questions for Chloe, please type them in the Zoom Q&A window. We're going to have time for a couple of questions right after Chloe's talk, and then the rest can be addressed during the discussion at the end of today's session. Chloe,
Thank you, Aviv, for the introduction. It is my pleasure to be here, speaking on the behalf of a large team to introduce you what we've done for the HCA Immune version 1 Atlas, which was really geared towards solving some key cell annotation challenges we have for immune cells. And this was done with members of the Immune Bio Network. As Aviv mentioned, this is a close collaboration with a genetic diversity bio network here with Shyam Prabhakar leading the way along with several trainees, and close partnerships with several members of the integration team. And have a family picture here of the team that has been, the training team that has been meeting weekly for well over 2 years to make this happen. And these are trainees that are affiliated with several labs, including Marta Lucan and Fabian Theis, Sarah Tagmans, Holger Heinz, and Prabhakar, Gary Reynolds, and myself. And we've done this in collaboration with several members of the entire Immune Bio Network. As I'm going to show you, we look forward to continue engaging with the full Bio Network as part of the Annotation Jamboree. So for those of you that are not familiar with our Bio Network, we have over 600 members across 40 different countries, and it's a Bio Network that I co-lead with my colleague, Dr. Gary Reynolds. The objective of our Bio Network is broad. We are seeking to map the human immune system across tissues, developmental stage, ethnicities, and lifespan. But immune cells are distributed across all organ systems. So a few years ago, we asked the question, where should we start? And to answer this question, we had several community engagement efforts where we led brainstorming sessions, and we ended up coming together with 4 tiers of priority. The first one being blood, because it's clear that's a window in human health. It's also the type of specimen where there's the largest number of single cell studies with direct translational application. The second tier is tied to tissues that are very immune specific, such as tonsils, bone marrow, cord blood, lymph nodes, and thymus. The third one being barrier tissues like lung, the GI tract, and skin that are really enriched for these resident memory, tissue resident memory cells. And finally, all our dirt tissues. And so we tiered them and ended up deciding to start with blood. And I'll expand a bit more on why blood. As I mentioned, it is a window in human health. It is easily sampled for research and diagnostics or with direct application to translational work. People are continuously profiling peripheral blood molecular cells, and that's why there's so many data sets out there. And so the idea here is that data integration could really empower future disease research. There's also the fact that having a really clear blood cell atlas would enable us to better decipher tissue-resident memory cells from circulating cells in the next phase of atlas building. So here we hypothesize that there are a finite number of biological important immune cell states and programs, and that some will be conserved across disease conditions. For this particular atlas, we focused also on integrating some initial disease. Our version 1 atlas includes 9 million cells across 2,600 samples and 2 conditions, and I'll expand on the 2 conditions in a minute. Our goals were to establish the detailed reference of circulating immune cell atlas, as I mentioned, to eventually be able to distinguish them from the tissue resident cells. Because we are the bio network with the greatest number of cells, greatest numbers of samples, we also had an opportunity to optimize the frameworks for atlas building at scale that could be more broadly applicable to other bio networks in future iterations. We also were seeking to generate data sets that would empower us to bring together the community to cell annotation jamborees to derive consensus cell annotation for immune cells. And of course, we wanted to support queries related to the host immune response and how demographic background can shape cellular identity. This being done in close partnership with our genetic diversity bio-network colleagues. As Aviv mentioned, these are the 5 different criteria that the HT organizing committees use to approve atlases, and I'm going to show you how we've met these criteria. It was different driven by our 2 different bio networks. We've incorporated some key landmark studies. I'm going to show you how our atlas is robust to technical factors, and we're starting to move some of our notation on CAP, and how we're making our data available on the HT data repository. So these are the data sets that were being integrated here. They included both some unpublished and published data sets. And here we ended up covering both healthy and COVID. And there was a debate that was part of our outreach and outreach effort a few years ago in terms of which disease we should include. And we were divided into groups saying that we should just do healthy, some saying we should do at least 1 condition, some said we should do many conditions. And we ended up landing on starting with 1 condition. And why is that? That's because to assess a good integration, ideally you would want data sets on the same condition, in this case COVID, generated across multiple institutions, multiple independent studies, and ideally different countries, so we could really assess the robustness of our integration. And the COVID data set gave us that unique opportunity to build that foundation knowledge before we start integrating additional disease, which is actually already happening as part of our next iterations. We had data across different 10x genomics chemistry, and you can see that they were across a different sample size. Some had like 40,000 cells, some had over 2 million cells. Some had few donors, some had close to 1,000 donors. We ended up pre-mapping all of the FASTQ files to the same reference genome and collecting our technical metadata. As I mentioned, we covered normal and COVID, and some of the data sets we included had other conditions, which we kept in the object. We're almost 50-50 between male and female. And you can see that, as Aviv mentioned, we have one of the atlas with the broader ethnic representation, which was supported by the Genetic Diversity Bio Network. We do have some continents that are not covered, and in partnership with the Genetic Diversity Bio Network, we're hoping to be able to include more representative data sets in the next iteration. So for next few slides, I want to share with you some highlights of our strategy to assemble the V1 Atlas, covering some of our feature selection optimization, hyperparameter optimization, and how we approach annotation. And so we spent a fair amount of time here on optimizing the integration strategy to build a reference atlas that would be robust to technical factors. First, without going into great details, we spent a lot of time on reprocessing and defining the most optimal quality control strategy, including setting QC thresholds optimized across lineages, optimizing our feature selection, selecting highly variable genes in healthy COVID, and taking the union of both. We've actually tested 5 different integration methods and honed in on to harmony and SCVI, on which we did hyperparameter tuning, looking at both, for example, highly variable genes, PC and latent factors. We optimized integration method specific parameters and the size of the neighborhoods. To assess which integration strategy between harmony and SCVI performed the best, along with which parameters, we use both quantitative and quantitative assessments. So starting with the qualitative assessments on the left, we use both technical and biological evaluation. For example, looking at an embedding, we could look at the assessment of different technical variables, such as the source of the data set, the 10x chemistry, the batch, the cell range of versions, the reference genome, the gender, the donor, the number of genes detected, and percentage mitochondrial signatures observed. And so, for example, in this particular embedding, we could see some clusters that were very batch specific, in which case we rejected that integration. We know so much about immune cell identity that we can also use this information to assess the biological variables and look at co-localization of markers for non-contentious cell subsets. For example, here, we could notice that there was a lack of resolution between IgG and IgA plasma cells, which just split, in which case we would reject this integration output. We also use quantitative assessment here using different metrics, including some incorporating the SCIB pipeline, but we also developed some novel metrics using based on RNA index, looking at the co-localization of marker genes in neighborhood graphs. So the same way I told you that we know about some key marker genes defining cell subset, we can quantify this to see if some cells with expected markers would co-localize. So to give you an example here, with some CD4 T cells, you have the score on the x-axis across the different cell subsets. In orange are the SCVI output, and in blue, the harmony output. And we, in the context of harmony for the CD4 T effector memory cells, you could see some really nice co-localization leading to a smaller score, whereas SCVI didn't give us cells that really co-localize as well, giving us a larger score. So we could more systematically quantify this co-localization and then manually verify the information to kind of speed up our hyperparameter tuning. In terms of annotation strategy, we really wanted to go beyond cell lineage and capture trilineage diversity. And it's really tricky when you have to deal with so many cells. So we perform fine-grained splits within each lineage to better capture the biological variability across the millions of cells we've analyzed. And given the scale and the standard lineage level grouping can miss key interlineage diversity, it can also misalign cells, leading to inaccurate biology. We had to figure out a strategy to iterate quickly. So we ended up starting with splitting cells across lineage, removing doublets, moving misclassified cells between lineage, and iterating this until we got to specific non-contentious buckets, which I'm going to tell you about. We ended up defining 33 cell subsets across CD8 T and NK cells, 25 subsets across the CD4 T cells, 27 subsets across myeloid cells, and 31 subsets across the different B cells. And these are the buckets we ended up landing to do unbiased clustering. So for the CD4, for example, we split the naive and central memory from the effector and proliferative cells. For the CD8, similar split, but we also included the gamma, delta, NK, and mate cells. For the myeloid, I'm going to show you a little bit more, but we did dendritic cells separately. For a naive, for plasma and B cells, we did the B cell naive memory in plasma. We definitely captured some hematopoietic stem cells and kept all the cells in a different bucket, those that didn't fit anywhere, in case some of you would be interested by these cells. And so showing you the myeloid cells here, if there's any dendritic cells aficionado listening, we did do two different splits where in some cases we kept the CDC2 cells with the monocytes because we know they share some gene programs with activated classical monocytes, so we could define this nice trajectory and also look at the dendritic cells separately, defining 16 subset of monocytes and 11 subset of different activated DCs. For every one of these cells subset defined across lineage, in addition to defining broad marker genes, we also make sure that the proportion of cells per cell population here, with every bar representing one of these cell subset identified on the left, were normally distributed across disease, assays, and studies, which you can see here. Every condition is well represented. We did this effort across every lineage, including the T cells, and just to kind of give you a sense of the different embedding, we had 12 subset of naive and central memory, 10 of effector, and 11 of NK, gamma, delta, and MAIDs. And all of these annotations are currently being finalized and are going to be uploaded for CAP to gather your feedback. For the CD4, we identified 10 subset of naive and central memory and 15 of effector. And you can see the global annotation projected on the left. And finally, for the B cells here, we subcluster these three compartments and got 9 subset of naive, 14 of memory and atypical, and 8 of plasma, all of which are currently going through a final phase of annotation and will be posted on CAP in the coming weeks. All of our code has been compiled on GitHub and will be made available. We also are making all of our FASTQ files available in the HA data repository, along with the source data set, our integrated object that will be also in the HA data repository and eventually deposited also on the Cell by Gene portal. What we're also doing is, as I mentioned a few times, moving our data on the cell annotation platform, which Aviv mentioned a few times. To give you a sense of what this looks like, I'm showing you a recorded video where we've uploaded our data in private space for now for the myeloid cells. You can see here the list of different subsets I was just showing you that are color-coded across different bar graphs here for the proportions. You can explore the data set. You're going to get a summary of the study along with some of the metadata that were collected. You can go on the left and select download data sets if you want to get so the end data file. You can click on molecular data. It's going to upload the embedding I was just showing you. Along on the left with the full list of cell subsets that were defined, along with the number of cells for every one of these subsets. You'll be able to click on these subsets and give us feedback, and we'll have separate sessions to explain to people how to do it. You will also be able to go and look for any of these cell subsets and get to see the annotation metadata that we've used to define these were unique cell subsets. You can look at our marker genes here by clicking here, and it's going to show you a heat map here and the key marker genes that were uniquely expressed in a particular cell subset that we've used for annotation. You can even click on this little icon here. It's going to show you the expression of this marker. And so with that, you can effectively navigate all of our data. And so standardizing the input is one thing, but now comes a real challenge where we need all of your help, those of you who are immune cell lovers. Now we're going to have the same data integrated with the same pipeline. And so I want to know, if I give you all the same 9 million cells, will you give us the same annotation? We're going to start everybody with a set of annotation that we spent a lot of time doing, and we're open for your feedback. And this is going to lead us to the Immune Bionetwork Jamboree, where you can either download our data or use a CAP user interface to directly give us feedback. There's going to be 3 proposed tasks. The first one will be mandatory, where we invite you to define preliminary cell subset identity and provide the parameters and clustering solution that you use along with marker genes. There are 2 additional tasks that are optional. If you complete task 1, you can actually provide a solution for cell spectrum characterization. We know all too well that cellular identity are not binary when it comes to immune cells. They're part of a spectrum. So we invite you to leverage our data to try to define landmarks and propose quantitative readouts of cell spectrum definition. We are also seeking participants to define the type of coverage for cell subtype recovery. The immune bio network is unique because we have so many cells, so we have an opportunity to assess how many donors, sites, technology, processing, and modalities were actually needed to identify all predicting cell subset within any particular cell lineages. And we're hoping that the output of such task would help help other bio network leading similar efforts. So stay tuned because we're going to be circulating the sign-up sheets this coming month, and we're hoping to be able to launch it by the end of this year. We will be giving 12 weeks to complete these proposed tasks. There'll be informational sessions coming up. The goal is to compile all the results early next year and to then schedule an in-person meeting for those that have participated so we can compare and contrast our results. So this is how we met all the key criteria. We're currently finalizing uploading all of our data on the HE data repository this month and are moving our orientation on CAP so that we're set to launch this Jamboree, which I hope many of you will be keen to participate. So with that, a last shout out to the amazing team. This has been a labor of love and putting this together to empower this community to work with us on annotation. A very big thank you to the entire Immune Bio Network and to the Czech Zuckerberg Initiative for us funding this integration effort. So with that, I'll stop sharing and we're going to continue the presentation here where we're going to tell you a little bit about HTA 2.0. So as Aviv mentioned, we're focusing hard on finalizing HTA 1.0, but at the same time, in parallel, we are also working on crafting the strategy for HTA 2.0. Next slide, please. And so HTA 2.0 is building on this version 1 atlas I just told you about. This is an overview. I'm getting even more detail on the next slide. And so we are moving from individual version 1 atlas to a unified reference. But as part of HTA 2.0, we're also trying to bring this data in a spatially resolved way. We want to make sure that our atlas is clinically applicable and that we leverage our high quality data to build foundation models. Next slide. And so what do I mean by that? So for HTA 1.0 version 1, the idea was never to have 18 independent version 1 atlas as the final product. Our goal is to next integrate these individual V1 atlases in a single unified reference that can empower the next phase of our atlas, including leveraging this to build macro panels and to try to better understand the biology in 2D and 3D through a spatially resolved atlas. We are now fully embracing an HTA 2.0 or disease program, which are the human in vivo perturbation models that can teach us so much about human biology, also show the clinical utility, and give us an opportunity to make sure that we build a globally representative atlas. And as we gather very high quality data sets and standardized file format and metadata, we have an opportunity to drive HTA 2.0 foundation models, build AI agents, and help support virtual cell endeavors that are being launched around the world. Next slide. And so what we've been doing over the last year is doing a lot of landscaping exercise to make sure that we tailored our strategy according to the needs of the community. And we've done this across 4 different tracks, and I'm going to quickly summarize. The technology track led by Holger Heinz and Jay Shin, the computational and algorithm development track led by Shyam Prabhakar and Oliver Stiegel, the product development track led by Niels Gettingsberg and Mazanifa, and the Atlasing Health and Disease track led by the Biological Network with my help for coordination. And each track was tasked with defining critical questions, challenges, opportunities that the community, our community, could tackle as part of our special program. And the output of these tracks is what's shaping our strategic plan. So I'll give you one summary slide per track, with the first one, the next slide, being the technology track. And so for the technology track, what they've been doing is trying to define the technological landscape, shortlist technologies that could be scalable to match them with the different biological network, and identify some key technological partners. So in thinking about features, they've been comparing capture versus imaging-based technologies, those that could accommodate FFP and flash frozen, the areas of the spot to make sure it could be single cell resolution, the type of marker panels that could be built, how quickly we could turn around samples to enable scaling up, how they would perform across many different organs, and if we could do 2D versus 3D. And so the goal right now is to test these technologies and work really closely with data contributors from the HA Bio Network to pilot these technologies in order to create a roadmap to scale up very rapidly in a way that can maximize accessibility to all and make the cost more efficient. Next slide, please. The second task force was focused on computation and algorithm development, and I should mention on the far right of these slides, you see the different members that have contributed to the task force. And here they want to define algorithm and analytic challenges that needs to be overcome to empower the spatial atlas. And so for the first step, they've actually looked into the optimal methods to optimize panels, thinking about algorithm tied to cell segmentation in QC, methods to integrate single cell suspension with spatial methods, and how do we integrate our V1 atlas with our spatial data. The second step includes going deeper and thinking about some of the analytical challenges we will face, including, defining cellular niche, thinking how we can integrate H&E staining to do digital pathology, for example, how to integrate multimodal data, and how to, for example, democratize analysis. And so they're currently working on a white paper to gather all these thoughts that will be key for fundraising, thinking about different hackathon, and working with the other task force to find the right companies to partner with to do these efforts at scale. The next slide, the third track has been focusing on portals. And so for portals development, they've been trying to figure out the best strategy to develop them. Next slide, next please. And so what they've been doing is reviewing the existing infrastructure, including what's being done to process raw, host raw and process data. How do you query spatial data? What are the best visualization output with analysis? Next slide. And to do so, they've been collecting user needs and review existing interface through different interviews. They've also been thinking about how to navigate metadata and how to visualize it. Next, please. And so with that, they're currently assembling an actionable roadmap. They've identified defining metadata specs as the key challenges that needs to be solved with the other task force, such as computational and data generation task force, and now to think about indexing analysis results. Next slide. And so finally, the last track is about how we're going to be generating some data. And so we've been brainstorming a lot about the scope of our minimal viable products for a spatial atlas. And it's clear that there's two paths. The first one is about considering deep profiling of tissue in a few donors. So we take large section of tissue in very few donors, but you profile deeply so that we can start reconstructing normal tissue in 3D. The second one is tied to case control study designs when you collect very small amount of tissue like a biopsy, but you can do it in hundreds of individuals, which is really key for capturing wider demographics and show atlas utility. And so we're going to be pursuing both. The group has also been thinking about the biological and technical metadata that are needed as a minimal product to be able to integrate our atlas. And so that's currently ongoing. Next. We've also been trying to think about additional measurements that needs to be done systematically on our tissue, for example, H&E staining, potentially whole genome sequencing, or specific protein staining. And next, and how we should think about building our marker panels. And it's becoming clear that the most logical way to approach this is to leverage our version 1 atlas that every bio-network is building, and to building on the cell annotation template where you're all invited to participate, where we're going to try to work towards consensus annotation and use these output of the template to build these harmonized marker panels. Next, some of the key challenges, and this is to be done in partnerships with different tech companies. And this, we've been working with task force number 1 in identifying these different technology companies that could partner with us to do this at scale. Some of the current challenges is that some of the initial efforts that are ongoing are focused on case control study designs. And some of these technologies don't allow us to do large regions of tissue to do 3D reconstructions, but there's a new technologies coming soon on the market that may help us overcome these challenges. Right now, it's mostly brain and the development atlas bio network that are tackling these issues. So lots for us to do. And so I'm going to hand it off to Shyam Prabhakar here because we had different calls to action here, and it's not too late to join the effort. If we don't hear from you, we don't have an opportunity to engage you in HCA 2.0. So I'll let Shyam tell you more about this.
Thanks very much, Chloe. The key thing I'll say here is we often hear at HCA meetings, how can I participate in the HCA? So this is your chance. One great way to get in at the beginning of HCA 2.0 is to contribute your samples to the data generation effort. And then, of course, you will be part of the data analysis effort for your samples as well, part of the science. So getting in right at the start, right? And that is the purpose of these two surveys we have here. One is a spatial focus survey. What samples are available for spatial omics atlasing all over the consortium, all over the world. And second is a disease survey. What disease cohort samples are available for the disease focus of HCA. And as you know, data generation is getting easier and easier in single cell and spatial omics. And cohort sizes are getting larger and larger. So the bottleneck really is, in many ways, the bottleneck is the samples, the cohorts, and the metadata and the consented metadata and so on, right? So please contribute to I'll show you the results from these surveys so far, but the surveys are not closed. And we really hope you will contribute your information to these surveys so we can expand it and you can be part of this effort. And I should say, you can be part of the effort even if you don't contribute samples. But yeah, this is one great way to contribute. I will skip to the next slide. We asked for various key kinds of information on the samples you have banked already for spatial omics or can collect at very short notice, like a few months. We have frozen and FFPE, but for spatial, perhaps FFPE may be the initial focus in terms of data quality and ability to get archival samples, retroactive clinical data, and things like that. Many of the respondents have generated spatial data already of their own, and that is very exciting to see the penetration of these technologies. Whole organ is obviously more challenging. Often people have only pieces of the organ or even biopsy. So 80% of the respondents had small biopsies, but a good number had fairly large pieces, like pieces bigger than 5 cm, which is great because, as you know, the spatial omics technologies are able to profile larger and larger pieces of tissue now. And you can get much more information on the architecture of a tissue from, say, a few centimeter resection, perhaps, or autopsy sample than you can get from a, say, a needle biopsy, a core biopsy, right? So, and then what resolution are people looking at or interested in? The great majority said they're interested in cellular resolution, and indeed, we listen to that feedback, and that is what we're going to prioritize in the Human Cell Atlas. But people also interested in subcellular resolution, and we cannot leave out regional either. There's substantial interest there as well. So that's the spatial results. Next slide. We also did a disease survey, again, very similar questions to understand what samples are available in tissue banks for disease analysis and what diseases people are working on right now, right? So that we want a ground up process for deciding the focus areas for HCA 2.0. And the results on the next slide. So even for the disease survey, we had more than 200 respondents. Fantastic. But again, please bring this number up to 300 or 400. We want more responses. Very happy to say we have responses from all over the world, not completely even representation of all continents still, but pretty good compared to say some other consortia. And we hope that all of these respondents will contribute their samples and will be part of the survey. And they've helped us focus on diseases of high global burden, as most respondents, but also local. We will not overlook local and regional disease burden, will not overlook rare disease. Because as you know, there's single cell and spatial technologies can give you a lot of information very fast on that steep learning curve for rare disease. Which disease areas? Metabolic disease and cancer, infectious disease are major ones, as well as autoimmune disease. So these are places where HCA members are interested. They have samples. They're already doing research in the area. And so these would be the most obvious disease areas of focus. Next slide. All right. Lastly, repeat the call to action that we've started out with in this meeting. For HCA 1.0, as you know, October 2026 is the 10-year mark. So we need to be very, very focused on finishing all 18 atlases by then. So please help us get there. Please be part of that, celebration, the 10-year celebration, by contributing your data and metadata requested by the bionetworks, and guide how we interpret the results from all of that analysis by joining the annotation jamborees on the cell annotation platform, so that we can get the community's input on what these cells really are. Okay, so finite deadline, October 2026, HCA 1.0, HCA 2.0, a lot of excitement in Milan and the general meeting in Singapore as well on the new directions on spatial disease and diversity, or expansion of all of these things. They're not completely new directions. So please, some of you are part of other consortia. Please help us, besides HCA, please help us connect with those consortia and coordinate our efforts, because there will be some overlaps always, and some shared interests and synergies we can identify. And then define the minimal viable product. We kind of know what a minimal viable product is for single cell. And indeed, you already have Atlas qualification criteria that HCA has been using. What would that look like for spatial? And then open source foundation models are exciting new vista. You've heard a lot about, you've heard of virtual cells from single cell data. With HCA 2.0, we will be in a great position to extend this model to, let's say, virtual tissues from spatial omics data, and understand disease as a perturbation, as well as siRNA or CRISPR as a perturbation. And finally, as indicated in the surveys, most immediately, please notify us of your cohorts and samples that are ready to analyze in HCA 2.0. Hope to have you all with us on the closing out the first 10 years and launching the next 10. Thank you.
Thank you, Shyam. And perhaps before we add concluding slides, I realize we don't have questions. We invite you to ask us questions, but I can perhaps start by asking you, what are you most excited about HCA 2.0 as we try to plan the next phase? It's a broad question because there's so many things, but what are you most looking forward to, Shyam?
Can I say all of the above?
I think we have a really, no, well, yes, you can say all of the above, but when I tell people how we're approaching this a little differently, given that we need to build upon 10 years of amazing work and leadership from Aviv and Sarah, and how we can solidify the reference atlas requires us to centralize some of the effort is on this through these lens, Is there something specific, a specific call to action you want to further highlight?
So I do think spatial is an amazing opportunity, and we can differentiate ourselves from existing consortia because we're coming in at the right time. Spatial omics technologies are maturing and improving rapidly. They're becoming more robust. The timing is really perfect for HCA to make an impact in spatial and define atlases. I don't know of any widely used or comprehensive spatial atlases right now that are publicly accessible. So there's a lot of work HCA can do in that field. But I do want to tie this to the fact that these spatial technologies can accommodate FFP samples, means that we can access archival tissue blocks from hospitals and make a major impact on disease. So I think those two go hand in hand. And of course, the portability of these tissue blocks means that even if you don't have the spatial technology in your city or in your hospital, you can ship it somewhere else and still be part of that activity, still be part of the science and the discovery, means that this gives us an opportunity to expand diversity as well in the HCA.
Thank you, Sam. And perhaps I'll invite Aviv to tell us, what she's most excited about this next phase, and perhaps your personal call to action before we move to conclusions.
So I'll iterate maybe one thing at least that Shyam said, which is the spatial component has always been part of the HCA plan from the very, very beginning. You can open the white paper, it's there. But I think it had a longer gestation period than we originally anticipated. Mostly, I think, for technical reasons, that technologies had to mature and be sufficiently scaled, accurate, robust, etc. And they're totally there. And I think as a result, we can actually move a lot faster in the spatial domain that we have in our previous years, because we kind of have all the pieces. We know the organs and the tissue, and the people are engaged, and we know sometimes we even have the specimens already, like everything is ready to go. And so one can do a very coordinated and effective effort to get very far very quickly. And I think the value of the data is enormous in this specific case. It's also true that for many spatial technologies that rely on FFPEs, you can actually deploy them more easily in more sites than the old days of single-cell RNA-seq from fresh tissue. So from that point, it's extremely exciting. The second area that really excites me is the opportunities with AI and foundation models, as well as with agents. With foundation models, we will never measure all cells, right? The whole point of the atlas is that it becomes a general enough resource that it kind of fills in its own blanks. And that's actually what a foundation model allows you to do. So it's one of the few areas in biology that you have really big data available today. That's why so many people are drawn to it right now. And we have a huge opportunity both with the first generation data because of how well the atlases were done, and with the next version of the atlas with the spatial component more prominent, to really empower the building of foundation models for cell and tissue biology. And then related to that, I find the agents really exciting because we do know that a lot of the barrier in extracting biological insight and knowledge out of large-scale data, even nicely annotated, organized, integrated, etc., is actually the analysis that comes on top of it. And this is where autonomous agents are becoming more and more empowering for biologists and clinicians and others to really engage with data like that. So that is also very exciting for me.
No, I hope you share all of their excitements. I certainly do share all of their excitements. We're far from done. I hope you're getting the message that we're far from done with our HC endeavor. We're only starting, and we have built this amazing community that you're all part of, and we really hope you're going to engage with us in pursuing HC 2.0. And Aviv, I'll let you lead the concluding words.
Sorry.
I'm happy also to assist here.
No,
So
I'm glad to close. I was just
okay.
taken for a moment by slight surprise. I didn't
Sorry
watch
about
the
that.
time very closely, so I didn't realize we're already closing. So
Yes.
I just want to first of all remind everyone how to be involved. We have the membership hub. It's a platform known as HCA Hub. And there you can search and connect with other HCA members, but of course people can only search for you if you actually sign up. And you can do it based on location and your research interest or your academic affiliation. You can look at job opportunities as well as post jobs, and you can participate in different interest-based discussion groups. I think the main thing that was always true about HCA is that it's really up to you. The barrier to entry is very low, and you're very embraced for everything that you want to contribute. So we really hope people will continue contributing and engage in new ways for this next generation of the Atlas. And I think with that, I will thank the HCA Executive Office staff, Aya, Yoshi, Esther, Bridget, Ann, John, Lucia, Christine, Ellen, and Samantha for all the work that they do every day to make the HCA possible, and our commercial partners in the discount program, 10x Genomics, BioLegend, BioScrib, Complete Genomics, Element Biosciences, MGI, Parse Biosciences, PixelGen, Nanopore, Oxford Nanopore, sorry, Scale Biosciences, Single Ron, and VizGen, that really allow HCA members to use all of these technologies at a discounted price. And finally, there are surveys that help us inform the strategy planning, the spatially focused survey and the disease focused survey. Please participate in the survey if you have not participated in it already. And it is 6:59, Chloe, in my time zone. And I
Yes,
guess 9:59?
that's right. AM and PM, depending on where you are and which coast.
Yeah, and for Shyam it is 10:59?
10 a.m. Good call.
10
10
a.m.
a.m.
So 9:59 for both of you, but not the same AM and PM, and 6:59 for me. And that's really a nice way of closing the HCA because we're truly a global organization.
Thank you all for joining, and we look forward to engaging with all of you in finishing the version 1 Atlas and launching HC 2.0. Good to see you all.
Bye, everyone.
Yeah.
Mm.